using your laptop?
Huh? Yeah, unfortunately. Okay, we're actually going to start out with some problems that will be relevant to Quiz 2. So pull out your clickers and think about that intestinal epithelium that we talked about last time. That's where these questions are going to come from. And there we go. Sorry about the late start. That's my fault completely. All right, so just down just a little bit. Okay, so there, and hold on, this can be open. Okay, so let's do this first one. Go ahead. I mean, if you have some paper, draw a little sketch of that intestinal epithelium. You know, Bart Simpson facing the blood and the lumen and blah, 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 and put the transporters that are involved in this question, which are SGLT1, GLUT5, GLUT2, the sodium pump, where they're supposed to be. Because you'll need to do that in order to answer the question. So let's do that first, draw a little picture, and then answer the question on your own, okay? Go ahead and answer it individually, so I should be seeing numbers running up there. Clock on you guys. It's 3. Okay, that's a good, nice consensus. So we may not even have to do the group stuff today on that one. So cytocalasin B, why doesn't that inhibit glucose if you put it on the atypical side? Which transporter is it going to block, if any? On the atypical side? GLUT5. GLUT5 transports what? Fructose and not glucose. That's why glucose is not blocked. And so the correct answer is the basolateral side only for that. Okay, so that's good. That indicates to me that you know what you're talking about. So let's do this one. Yes. Right, so it blocks GLUT2, which is only on the basolateral. That's why putting cytocalasin on the basolateral. So if you had a patient that you wanted to do this to, heaven knows why, you want to give them diarrhea for just for laughs, you know, you'd want to give it to them IV and not through the mouth. Okay. Okay, so let's try this one, number 2. Okay, so we have sort of a split decision between answer 1 and 2, and so let's look at that. Luminal, well, those are the two sides. So talk to your group. I'm going to reopen the polling, and let's see if we can come to some sort of consensus. Open. Are you in a group back there? Red shirt? Where are you? Get with your group. Come on, let's talk to them. They can't be that bad. Ten seconds. Okay, got all 18. Good. And 21 groups in here. Wow. You're multiplying like rabbits. Okay. Answer 1, it seems to be we're coalescing around there. And so why is that the case? So the answer there is if you put it take it away from the luminal side, that would block glucose transport. Why is that the case?
So you think like this transport are coupled on the apron
And not on the basolateral, right? So what if you took it away from the basolateral side? What would that do? Sodium. And we're osmotically replacing it with some. When you do a sodium replacement study, usually you have replace it with something like choline so that you don't have an osmotic gradient across the epithelium. But let's say we did that. No sodium on the basolateral side. Why wouldn't that block glucose? Somebody else? Well, if the answer to inhibit it is it's because sodium and glucose transport are coupled, so if you take it away from the basolateral side, the answer must be something about sodium is not coupled to glucose transport on the basolateral side. And so that's the way you should think about that. And so the answer to that is sodium transport through the pump happens just fine in the absence of sodium on the blood side because it's transporting sodium from the cytoplasm into the blood. And it just helps it that much faster if there's no sodium in the blood, right? So it's not going to prevent it from transporting sodium into the blood. Is it going to get sodium from the lumen? Yeah, because you left sodium on the luminal side, and so the SGLT1 is going to pull sodium, 2 sodiums, 1 glucose into the cytoplasm. The pump will kick it out into the bloodstream. It will rip potassium from the blood and put it back into the intestinal cell cytoplasm. So glucose transport operates just fine if you remove sodium from the blood side, but not the apical side. Okay. And this last question is related to that. And I think I killed my clicker, but that's okay. We'll do that without clickers. Would it inhibit glucose transport if you remove potassium from the blood side? First of all, you couldn't do this in vivo. The person would be dead. But this is in an isolated system. You've got the intestine and some little transport apparatus that you're looking at.
Are you supposed to?
No, let's just, who has an answer? Raise your hand, somebody. Don't be scared. Somebody that hasn't answered yet today. Yeah. Exactly. Right. So if you prevent the pump from binding to potassium, you can't go from E2 back to E1. That's just like blocking the pump with Guavain. So what does that do? Well, what that does is the sodium gradient, the level of sodium in the cell goes up, right? And what's that going to inhibit on the luminal side? SGLT1, because it runs off the sodium gradient, which you've just allowed to dissipate. And so indirectly, blocking that pump will block glucose transport. Okay, so you need to think how about all these things are interrelated, and some of them are not. So that's the kind of game you need to play with this system. So that's good. All right, so now let's go back. We were talking about different types of ion channels. And so we talked about the classic one up here was this bacterial channel, but that kind of channel is not in us. Let's talk about one that's related to that channel, and that would be the voltage-gated potassium channel, voltage-gated K channel. That's what it's generally called. And let's get rid of this thing and get the PowerPoint. Okay. Okay, so we were talking about this bacterial channel, and this is a nice model for all the mammalian channels because this bacterial channel has 2 of the 6 different transmembrane segments that each voltage-gated potassium channel will have in you and I. Okay, so the sort of the same model applies. There's some sort of selectivity loop. You actually can. How do you close that channel? The inner helix actually closes up like a diaphragm on a camera. Okay, so what happens is back here, the way you block this, the way you block that pore is for this inner helix to squeeze against that one. And remember, there's 4 subunits. So there's one behind that little blue thing. There's one in front that have been removed for clarity. So remember, there's 4 subunits. But here's 2 of them that are shown. And so if this thing squeezes this way, and this S6 squeezes on the inside, you close the channel. This thing does not close because it's too stiff. It's meant to always be ready to accommodate sodium or potassium. You don't want that thing to open and close. You want this thing to sort of kneel inwards. Okay? So that's how that thing is gated. And so that's what this diagram shows, is this going from closed and opens out a little bit, and then it can go and close again. What actually gates this? It's actually changes in pH, and your book doesn't talk about that, but changes in pH will actually increase pH, will open the channel, decreases in pH. So decreases in pH, increases in proton concentration will open the channel. If it becomes more basic, it is you lower the hydrogen ion concentration, then you close the channel. So it's an ion gating mechanism. So let's talk about, so this is a picture of one subunit of the voltage-gated K channel that's in you and I. And we find that in our neurons. All kinds of neurons must have this protein. Okay. And so notice that here we have this S5 and S6 that are related in sequence, actually, to the bacterial channel. You see a difference, though? Look at the selectivity loop. Instead of having a poor helix, it's still got a selectivity loop right there, but what is it between? What motif or what secondary structure is it between? 2 beta strands. So this is actually a beta sheet that's holding the selectivity filter in place instead of a poor helix. But still, you have S5 and S6 is where that intervening loop comes out of. It's just held in place by this little beta sheet. Okay? And so this is going to form one side of that coordination that's going to grab onto the potassium ion as it moves through the channel. Okay? But that can't happen unless this pore is open. Okay? So something is going to gate that pore open, right? And that something is voltage. And we actually know which part of this subunit is sensitive to voltage. It's actually S4. S4 is the voltage sensor. The reason it's the voltage sensor is that it has positively charged amino acids, lysines and arginines, every 3 or 4, as you go through here, every 3 or 4 amino acid residues are positively charged. So you've got, this thing is bristling with positive charges. And so what's going to happen is, let's see if there's a good diagram here. Yeah. So in the resting state, the outside is positive, and so that actually pushes the S4 in the closed state closer down to the cytoplasm, so that S4 closes up the pore, right? Okay. Now what happens is, if you depolarize this, that is, this suddenly, the outside suddenly becomes more negative, as in this case, what it's going to do is the S4, it actually rotates 180 degrees and tries to migrate up to the surface. It does quite a dramatic change in conformation. And so what's going to happen is, as that S4 segment, it's one of these guys. So this would be, this is S6, that's S5, S4 somewhere. It's going to rotate up and it's going to pull open the channel in response to a depolarization voltage. Okay. So that's why we mean it's voltage gated. So this guy, the thing that's doing the sensor work is S4. So that's the thing that's being ripped up towards the extracellular space that pulls open the selectivity, access to the selectivity filter, and then potassiums can move through. Okay. All right. Now, it's a little bit more complicated because you have this little thing. On the N-terminal, let's go back to the simple, looking at one subunit. So this thing, just like the bacterial channel, has 4 of these proteins. It's a homotetramer. Okay? It takes 4 of these to line the channel. And they all have a little ball on the N-terminus. Okay? And so you know, if the N-terminus is cytoplasmic and you have an even number of helices, then that's going to be, the C-terminus will also be cytoplasmic. This thing is called the inactivating particle. Okay? There are 4. There's one little ball for each one of these subunits. So in this position, they've cut away 2 of the subunits. Each one has a little ball, and they all try to compete to plug that hole. Okay? So when does that thing swing up in there? Let's look at it. So this is the transport cycle. In this state, the voltage is polarized. It's not depolarized. It's highly polarized. And so it's negative on the inside, positive on the outside. That propels the voltage sensor, so it stays in place. And the thing is closed, okay? But in this state, it is closed, but there's no ball in here, so it's actually closed and activated. It's ready to go as soon as you open that channel. But it is activated. That's because there's no ball plugged in the hole. Okay. Now what happens is when you depolarize that, you go from this situation to that situation, where it's more negative on the outside. That jerks on a S4, okay, that opens the gate, and because this is activated and this is now open, that's when potassium can flow through there. So 2 conditions must be met. Number 1, you have to have the right voltage to tug on S4, which is the voltage sensor. That opens the gate. And you must also be in the activated state, which is no little ball stuck in the hole. Okay? Now, what happens is, when this thing does that conformational change, this thing opens. And what that opening does is it slowly starts this ball swinging into place. So one of those 4 balls comes flying in towards this pore, and what happens is after a delay, the ball swings up into the pocket, And it goes from being open and activated to open and inactivated. So the flow of ions stops, because even though this is still open, it's gated open. But it is inactivated by one of the four balls. They all compete to try to get in there. And it was the initial confirmation change of the gate opening is what started the ball moving. You ever played that little game with you have a little cup and the ball's attached to it? Try to catch. That's exactly what's going on here, right? So you start to move your hand, and it takes a little while for the ball to kind of come up over and try to catch it on the lip or inside the cup. But that's what happens. The movement of this ball is much slower than the opening and closing of that gate. Okay, so this channel, voltage gated channel, has three states, right? It's closed and activated. It's open and activated. That's the only condition through which ions can move. And then it is open and inactivated. No movement. Because you got to have a pathway. Anybody, any NASA growing physiologists in here? Why do you want to inactivate the sodium channel? You'd have to really know what causes impulses to go up and down the neuron. It actually prevents your nerve impulses from going the wrong direction. The nerve impulses need to go in one direction in order for your brain to work properly. Because if they went like this, what would you do? We'd all be a puddle of cytoplasm. They have to go unidirectional. And so this prevents nerve impulses from reversing in the wrong direction. Okay.
How do you get the ball out of the metal?
When this closes, that conformational change causes it to drop it. It doesn't like to be stuck in there with the tube closed. Okay, good question. All right, so now let's go on to a different type of channel. That was voltage gated. We've talked about pH gated. That's sort of an ionic gating. Ligand gated. This is an interesting topic. These are ion channels that are gated by some sort of chemical. And so what we're going to use as our poster child is the neuromuscular junction. Okay? So you have a nerve that's coming in here, and it's innervating this big thing right here, which is a skeletal muscle fiber. Okay? So we call where this nerve kisses the muscle cell the neuromuscular junction. Okay? So the nerve cell is going to release a neurotransmitter. Let's take a look at it. Okay, so that's a cartoon of that's what it really looked like on the other thing. This is a cartoon of the axon terminal. Okay. So what's going to happen is you're thinking, I want to move my hand, right? Okay, so your brain sends an impulse through nerve cells, and those nerve cells carry an impulse, which is just a change in depolarization, and that action, it's called an action potential, and that action potential reaches the end of this, and it opens not a voltage-gated potassium channel, but it opens a voltage-gated calcium channel. Okay? And it looks very much like a voltage-gated K channel. But when you open that calcium channel, calcium pours in and tells these vesicles that are full of neurotransmitter to fuse with the axon terminal. So you can see that's what's happened. This thing just moves up against the membrane and fuses. Okay? And when it does that, it's going to release the neurotransmitter, which happens to be, in this case, acetylcholine, a real small molecule. And since this is sitting right on top of this is the muscle cell, right? This is the target. The muscle cell is sitting right there. This goes very quickly across that little narrow space, which is called the synapse. And so the acetylcholine diffuses, and it binds to this channel. This is it's a type of cholinergic channel. Why do we call it cholinergic? Cholinergic because it's activated by acetylcholine. Okay. And you actually have two different flavors of cholinergic receptors. Okay. So the family is cholinergic receptors. That's because acetylcholine, which I'll abbreviate ACTH, is the thing that's binding to this family of receptors. Now, the receptor is on the target tissue. The target tissue in this case is muscle, skeletal muscle. Okay? And this cholinergic receptor, there's two different types, two different flavors. There's the type that responds to nicotine. Okay? Nicotine is an agonist for your neuromuscular junction. And so we call it the nicotinic cholinergic receptor. Okay, nicotinic. That's the one that's on skeletal muscle. And a number of other places. Okay? Now, the other flavor, and there's actually several different proteins in this class, it's called muscarinic. And that's because the mushroom chemical muscarine activates that class of receptors. So there are two different classes of receptors. So we're going to deal with this one right here. Okay, that's the one we're looking at. Okay. Now, what's interesting is the receptor is itself an ion channel. The receptor for acetylcholine that binds it is going to use that ligand to open and gate the channel. Okay? Everybody got this? All right. So it's going to bind to the receptor. This is the nicotinic cholinergic, the nicotinic receptor, let's just call it that. And those two ligands bind to, it takes two of them. Okay? And so this thing oscillates in between an occupied and closed, occupied and open, and then after a certain amount of time, it goes ahead and closes even if acetylcholine is still there. And eventually the acetylcholine will dissociate and it's destroyed by enzymes that break it down. Because you don't want to leave the signal on too much, otherwise you will just be shaking all the time. You want to be able to very precisely control your skeletal muscle movement. So very quick signal to move your muscle and then shut up. You know, very quick signal. Okay, so let's look exactly how this ligand gates the channel. The channel itself has 5 different subunits. So it's a heteropentamer. Heteropentamer. You should know what that term means. So each one of these subunits contributes one alpha helix to the pore. One alpha helix. Kind of like it contributes one of the S6s right there to the pore. Okay, so the pores down through the middle of this little crowd of subunits, and this is what the alpha helix looks like that's on each one of those subunits. It's got a pretty similar characteristic. That is, it's got negative charges on the exoplasmic, extracellular space side. This is the synapse right here where the acetylcholine is coming in. On the cytoplasmic side, it's also got negative charges. What are those for? Those are to attract only cations. That's what makes this a cation selective channel. Remember, we saw the same deal for the bacterial potassium channel, right? But they were only on one side in that particular bacterial channel. Here, you've got negative charges on both sides. And this thing is fairly non-selective. It will take potassium, sodium, or calcium. So it's pretty, it's got a pretty loose in terms of its affinity. But what it does require is it must be a cation because of these little negative charges. Anions will be repelled. They cannot go through the pore. Okay? So what makes this cation selective is those negative charges. And then what is the nature of the gate? The nature of the gate are some hydrophobic leucine residues on each alpha helix that project into the pore. So in the absence, if there's no acetylcholine, these leucines are going to occlude the pore. They will not let water or ions through because they're hydrophobic. So that's the nature of the block. Okay. Now, so what we have to do is pull that apart and open the pore. So we need to actually move those lysines from here into this position. And what happens is acetylcholine comes and it binds right there next to the alpha subunit, actually the junction between the alpha and gamma subunit. Two molecules have to bind. And what happens is you get this picture. We're going to focus on just the alpha subunit. What happens is in the blue is its position when there's no acetylcholine. When it's in the blue state, those leucines are in the way. Okay? And when you bind to acetylcholine, it turns red. Not really, but it shows you that it's moved from here to there. So it moves out of the way, it opens the pore, and then something is going to go through there. That something tends to be sodium coming from the synapse into the cytoplasm. That's the chosen one. And I'll let you think about, you can't answer that question right now, but why is it sodium that goes through preferentially, even though potassium, sodium, and calcium can all move through that channel? That's a difficult question, actually. I think it's actually one of the set of problems. So we'll ignore that. But what does preferentially go through is sodium goes through here. So what's going to happen to the membrane potential of that cell if you've got sodium pouring through that hole? It's going to depolarize that membrane, right? And that when your muscle cell depolarizes, what's going to happen is that's going to lead to a release of calcium inside the muscle cell from the endoplasmic reticulum, which muscle people always call sarcoplasmic reticulum. And that's going to release the calcium, and that's going to cause muscle contraction, skeletal muscle fiber contracts. Okay, so that's the whole story. All right, is everybody clear what's going on? You get two different cells, and they're both being activated because the different channels are opening and closing. But this one is a really good example of a ligand-gated channel. Okay. Good. Okay, now, so we've actually got all the equipment we need right now in order to understand what generates a membrane potential. And I think if you don't get this, you'll be confused for the rest of the lecture. All right? So let's understand what causes a membrane potential to arise in the first place. And you have a link to the University of Toronto animation site. That's a really good place to look at this, because let me set this up for you. So here's a membrane, and on each side of the membrane, there's an equal number of negative and positive charges. That's called macroscopic electroneutrality. In general, all over the cell, there's macroscopic electrons. So there's a positive charge for every negative charge and a negative charge for every positive charge. It's only when you get close to the membrane and somehow a few positive charges migrate across is you start to get charge separation. So we need to ask ourselves, how do we move a couple of those positives across that really generates the difference in electrical potential? So the website goes through that very carefully. And so let's just look at it real quickly. I think I can have it. Give us just a little bit. So go to chapter 4 is resting membrane potential. And if you already know this, then forget about it. But so what we're doing is here's the cell membrane. We've got a voltmeter that tells you what is the membrane potential with respect to the outside. The outside is 0. Okay, so this is going to change with respect to the outside. And so we'll go, then you go through the pages here. Okay, so let's create concentration differences, because that's what happens. Some sodiums move from one side to the other. What did that? All right. So what's doing that is the sodium pump. The sodium pump's moving a few more positive sodiums out than it's bringing in, right? It's moving, let's do it again. It's moving 3 sodiums out for every 2 that comes in. So that's going to make the inside become a little bit negative. And you notice our voltmeter is now negative 55. All right? And it doesn't take very many moving to generate a fairly large membrane potential. Okay? And then once you've moved those, now you get passive movement. What's happening is you generated a higher concentration of potassium on the inside, so now you've got a potassium gradient that wants to go out. Where does it go through? It goes through potassium channels. And when you have positive charges leaving, what's going to happen to the membrane potential? It's going to go from minus 55 to minus 70, because it's going to hyperpolarize, because you've got positive charges leaving the cell in response to the potassium gradient that you just built up. So you've got more positives out here than you have inside. And so that's beginning to generate a membrane potential. So, and this is, you've already know how this works, so we'll just leave it in here. Okay, so that's what's setting up the beginning of the asymmetric distribution of sodium and potassium. And then what follows is then potassium moving passively through a potassium leak channel that's always open. Okay? Okay, so K flows out of the cell. And what happens is that attracts more negative charges to the membrane because there's more positive charges here. And this membrane is so thin that that electrical field can be detected across that space. And so what happens is these accumulations of positive charge attract these anions. Your cytoplasm is full of negatively charged anions. Most proteins are negatively charged. There's lots of phosphates, there's chloride, there's lots of stuff that will stick close to this membrane to neutralize that positive charge. Because this needs to be electrically neutral as well. Okay? So that's what it's attracting, those anions. So the more K that flows out, the more anions you bring to the membrane. So look at the potential gets more and more negative as you draw more and more ions to the plasma membrane. Where are they being drawn to? The positive charge on the other side of the membrane. All right. So now we have, at what point will, so potassium's built up inside the cell because of the sodium pump. When will it stop moving out? Well, it'll stop moving out when it reaches some sort of equilibrium. And at some equilibrium, what's going to happen is the number of positive charges that's moving out equals the number moving in. So that means it's in equilibrium. When does it reach that equilibrium? It reaches that unique equilibrium at the correct concentration gradient and membrane potential. So those two things have to come together to determine where that potassium equilibrium potential is. And so we're going to actually calculate that, right? Because we want to know when the flow of ions stops across this cell. So I'll leave it to you to go through onward with this and convince yourself where the membrane potential comes from, all right? But now what I want to do, so are there any questions on this so far? So we've clearly generated some sort of membrane potential. Usually it's, why is it not minus 85? That's pretty negative. That's because there are a few sodium channels that are open as well as potassium channels. And so if you've got that much negative potential, that's going to suck some of those sodiums back into the cytoplasm. If you just left it like that, eventually you would dissipate the entire sodium gradient and the cell would die. So what keeps it, what maintains the sodium gradient? The sodium pump is always working against this leak. The leak is much smaller than what the sodium pump can handle. The sodium pump can handle it, so it just keeps pumping them back out every time. It's just like bailing water out of a boat. And that keeps the membrane potential, it causes it to move closer to minus 70. What I told you is minus 60. It doesn't matter. It's minus 60 or 70. It's the balance between sodium coming in and generating a positive potential and potassium's leaving and generating a negative potential. Okay. Okay, so let's take a break because it is actually 10:15. And we'll use the Nernst equation to calculate things. There it goes. Yes.
What channel is the clicker on? For some reason mine
41.
stopped working.
So hit that guy,
Hit that.
and then let it, okay, now hit 41, and then hit that again. There, you got it.
Oh, okay. All right.
It's not like a big deal. It's not like
Okay, I got lost in your talking about the nature of this game.
Okay.
So you mentioned license and losings. What was it when you said again?
This one, they're not licensed, they're just loosens.
Just losing. Okay.
It's think about it, that's a really good hydrophobic plant.
Okay.
And so that's, when those are in the way, no cations can move. So what you do is you just jerk those out of the way.
Okay.
And what causes the thing to jerk is acetylcholine.
Okay.
Yeah.
Thank you.
What caused the ball? Okay, so what happens is when that channel starts to close, and when it detects the voltage, S4 pulls, and that causes a confirmation change. And when it opens, it's actually pulling on the ball real slowly. And the ball moves slowly so the ions can move for a couple of milliseconds and then it closes. The ball moves slow. It's kind of like that game. Your hand moves first, right? Then the ball comes. It's a confirmation sheet. And since they're all the N-terminus is connected to all the transmembrane segments, tension generated somewhere else is going to pull the ball. Well, when the ball goes in there, it stops the ion. In fact, what has to happen for it to reopen, the ball drops out because when it goes from open back to close, when that S4 comes back to its normal position, it closes the gate, and that causes pulls on the ball so it pops out. Okay, so you're going from the channel's open, right? Because the voltage is there. And when the ball comes up in there, it sticks in there. So technically the state is open and inactive. It's open and inactive, so that stops the flow. But what happens next is the voltage repolarizes so that the action goes on top. And what's going to happen is the voltage is going to go back to normal. S4 will roll back into its place. That will that closes the channel, and closing the channel boots the ball out. It's like squeezing it. It's like squeeze the cup, the ball. And now it's closed, but it's activated. It's ready for the next cycle. So the only thing you need to do is shift the voltage and it'll be open and activated. That's what you're doing. Yeah.
I have a question on the cutoff for the next course. Where is the material cutoff?
You're gonna see it. It's in bright red letters.
Okay, so
It's
we haven't
after
done it yet.
we're gonna get to it.
Oh, okay, good.
We're approaching it. We just have a
Okay.
few more. I have to teach you how to use the Nernst equation.
Okay.
Okay, let's get started again. And I did want to point out to you, I'm not trying to be sneaky, there's another tool you need to add to your toolbox, and that's this little chemical right here, tetraethyl ammonium, better known as T. You know, you can think of Tea Party blocks Congress, Tea Party blocks this potassium, voltage-gated potassium channel. Okay. So now, where we are is we want to figure out which way is an ion going to move if the channel opens. So there must be two conditions met for an ion to move through a membrane. Number one, there must be a pathway. Okay, so that means if there are ion channels there, that's okay, but they have to not only be there, they have to be open. Okay, so we're going to say, yes, we've got some sort of ion, and then a pathway opens momentarily. It usually stays open for a few milliseconds. All right. So that it has to be open, and then there has to be some driving force. It can't, if it's at equilibrium. Remember that point. At some point of concentration and voltage, potassium started going in and out at the same rate. That's the membrane potential at which it's at equilibrium. So we call that, strangely enough, the equilibrium potential. And so we need to calculate where that is so we can tell, is there a driving force that's going to push on the ion? All right, so it's real easy if there's no membrane potential. Okay, all you got to do is look at the chemical difference. If there's more outside than in, it's going to go in. If there's more inside than out, it's going to go out, right? So in the absence of a membrane potential, even if your ion is charged, you'll know which way to move it. Right? If the membrane potential is zero, because there's no pulling force electrically. The only pulling force is the chemical difference, and so it's going to push in or out depending on which is more concentrated. So that's an easy situation. And this is the same situation in a typical cell. It's negative on the inside, like -60. And let's say, what ion is more concentrated outside? Sodium is a good example. It's like 145 millimolar outside in your blood, and it's about 5 to 10 millimolar on the inside. So the chemical potential is pushing in, right? So if we open a sodium channel, will we know which way to go? Well, the chemical potential is pushing in. Which way is the electrical potential pulling? In the same direction. So we don't need the Nernst equation if both sources of energy are pushing in the same direction. So if the sodium channel opens, don't calculate anything. If there's a membrane potential, it's going to go in. And if there's a chemical potential, more sodium outside than in, it will have to go in. So if they're both going in the same direction, don't use the Nernst. You don't need to. All right? This is where it's complicated. And then the good situation that resembles this is potassium. It's higher inside the cell than it is outside. Okay? So the potassium, if you open a potassium channel, you'd say, well, chemically it wants to get out, right? There's more collisions on this side than there is on that side, all right? But the membrane potential is positive outside the cell, right? It's -60 on the inside, and it's 0 out here, so that's a positive potential that it's trying to move against. So that's going to impede the flow of potassium. Who wins, the chemical or the electrical potential? You can't tell until you use the Nernst equation to calculate where is the point at which these two things are balanced. What is the membrane potential which exactly counterbalances the chemical potential? And that's what the Nernst equation does for you. It'll take a given chemical difference and convert it into millivolts. And then you can ask the question, and I'll show you how to do that, which way is it going to move? So here's how you do this. Let's look at potassium. And so somebody get out a calculator. You may have a with log on it. Thanks. Okay, let's actually, before we do that, let's look at this equation that we're going to try to use. And that's shown up here, okay? And it looks pretty ugly. You know, it's the log of, so it's got all these This is the voltage difference at equilibrium equal to the chemical difference that it's balancing. And it's got a conversion from natural log to base 10 log. That's what this number is here. It's got the gas constant, the temperature, the valence on the ion that you're talking about, the Faraday constant. So that's a lot of jiggery-pokery that we can get rid of, right? So we can take this whole little thing and reduce this part of the equation to that number right there. So at room temperature, so temperature is a variable, okay? But we're going to say, I'm going to give you two values for you to memorize. One is at room temperature, this number is going to be about 58 millivolts. Okay? And that takes into consideration the gas constant, the temperature, and the Faraday constant. Now, the valence is still a variable that you need to change depending on the ion you're talking about. So don't make the mistake of doing this calculation if you don't understand what the valence is. So if it's potassium, the valence is, and it has a magnitude, +1, okay? So it's plus or minus, and then there's a size to it, right? What's the valence on calcium? +2, okay? So you need to feed that into that little part right there. So let me move this up a little bit. So when you're actually working with this equation, it's 58 millivolts, but you need to put the plus or minus in that denominator. See, it's 58 millivolts divided by the valence. So you take this number 58 and divide it by that valence for the ion that you're talking about. So if it's potassium, it's +1, so it'd be plus 58 millivolts, okay? And then what you do is you look at this number here. That's the concentration of the ion out over in. Concentration out over in. Remember that, out over in, out over in. You take that, you find that dividend, okay? And so you do the division first, and then you take the log of that division, okay? So in our case, if we're doing potassium, what we could do is, let's blank this out. Let's set up the equation. And so here's the cell, and this is the way you should do it each time, all right? So you have to be given what is the membrane potential before you open this channel. And we'll just say minus 60 millivolts. This is a horrible thing, right? And it is 70 millimolar of potassium on the inside. And it is, let's say, 5 millimolar of potassium on the outside. So you've already got the first part of your Nernst potential, right? So somebody calculate for me what that ratio of out to in is. It's going to be what? 5 divided by 70. Somebody do that. It's what now? 0.071. Okay. Now, take the log of that. So you do the ratio first. That's out over in. You take the log of this. What's the log? So it's going to be a negative number, right? Because the log of something that's less than 1 is always negative. Okay. So it's negative 1.15. Okay. Oh, it's 0.07? Okay, let's try it again. It's that? Okay, and that equals, that sounds better. Okay. So what you've got now is this part. You've got concentration out over concentration in. You've taken that ratio, you came up with this number, and then you take the log of that, and you came up with that number, which is a minus 1.15. That's correct, right? Okay. And now, what do you do with it? Well, you've got that number that's got all the constants, 58 millivolts. Now you divide it by the valence, which is +1, so it is plus 58 times a minus 1.15. And what's that? Minus 66 point something? 0.6. Let's just say, oh gosh. Park Avenue. All right. what that is, that's the Nernst potential for potassium. And I don't call it N, you call it E for equilibrium. That's the membrane potential will exactly counter. So if you open this, and the membrane potential is at minus 66.6, there will be no net movement. Potassium will stop moving. Okay. But that's not what our membrane potential is it? It's minus 60. So how do you tell which way it's going to move? Well, there's two ways to do that. Here's sort of the intuitive way. Okay. So what you do is there's your cell, and just draw you a line. It doesn't really have a Y value. So this is 0 millivolts. Okay. And we said our B sub M, that's the membrane potential, was minus 60. Correct? Now we just calculated the Nernst potential to be about minus 66. So that's the EK plus, right? So it's even more negative than where the membrane potential is. Okay? So here's the rule. If those two are separated, that means it's going to move somewhere. If they're the same number, it's at equilibrium and it cannot move. Okay, so that's the rule. That's the first rule. If there's separation, there's potential energy available to move it. And here's the rule. If you open the ion channel, and you can only open potassium at the same time. You cannot open anything else. You open the potassium channel, the rule is the membrane potential moves towards the Nernst potential. Okay? So if you open the ion channel, ions will move such that this collapses towards the Nernst potential. What's it doing? It's becoming, inside the cell is going from minus 60 to what? You're making the inside more negative. So the question is, which way do you have to push the potassium to make it negative on the inside? They have to leave the cell, right? Because it's a positive charge. And so if you open a hole here, the potassiums will flow out until the membrane potential equals the Nernst potential at minus 60. Okay. So that's the intuitive way. But a lot of people are not very intuitive with electricity. So I'm going to give you a different, and we're going to do this a couple of times. So I'm not going to. So the other way to do this is, let's say you want to just kind of have an equation in front of you. The electromotive force pushing on the potassium is equal to the membrane potential minus the Nernst potential. Okay? So let's see if that works. So what you do is you do this little. And so what that is it's asking, is there a difference between the Nernst potential and the membrane potential? Because that's the energy available to move the ion, right? So that's what it's asking for, is that separation. That's what this gives you, the electromotive force. And if you calculate that, it's what? Minus 60 minus 66. If you do the math, it's plus 6, right? Minus 60 minus the Nernst potential, which is itself negative, minus 6. That is plus 6 millivolts, and you take this number and put it inside the cell. Okay? So plus 6. And you have a positively charged ion being pushed by plus 6 millivolts. Which way is it going to go? It's going to repel it, right? It will leave the cell. Okay? Because positive charges, the same positive charges repel each other. And so that's plus 6 millivolts to push it outside the cell. So that's the other way to calculate that. Okay? Let's do another one. Just make up something. So let's do chloride. And I don't know what the actual numbers are here. In fact, we can get those numbers real quickly because there's a little table in your book. Let's turn this off. Okay, because this is the typical intracellular ion concentration. And so chloride is, let's say it's 5 inside the cell. So it's 5, and I'll put it back on. 5. What is it outside of the cell? 110. So now we got a problem. and let's say it's minus 60. So let's turn this back on. So I suggest you do the Nernst potentials for all of these ions based on these, and ask yourself which way is it going to move if you open a chloride channel, a proton channel, a calcium channel, and so on and so forth. Just give yourself some practice. Okay, so what we're going to do is we're going to open a chloride channel. Let me turn this off. We've got 5 millimolar inside. We've got 110 millimolar outside. So we've got 110 divided by 5. Somebody do the math for me here. It's around 20, isn't it? 21? What is it? I'm sorry? 22. Okay. 22, we take the log of 22, which is the log function. 1.34? Okay. So that's 1.34. Now, now we have, let's say it's still at room temperature, 58. What are we going to divide this by? -1. Okay? Don't forget that. And so this is now minus 58 times 1.34. What does that come out to? About 77, let's just say 77. Minus 77. Remember, there was a direction, okay? So let's do our little trick here. We draw this open, here's this. Put the membrane potential, because that was given to you. Now, the next thing you do is put the Nernst potential. It's actually more polarized, hyperpolarized, so it's minus 77. When we open this channel, we're asking which way is it going to go, right? And so the rule is, if there's a separation, it's going to move somewhere. It's going to move from one direction to the other. It'll be a net movement. So we know that's true. It will move. And it's going to, the rule is, when you open that channel, the membrane potential collapses towards the Nernst potential. In other words, chloride will keep moving until it reaches the equilibrium potential, at which point it stops moving. Okay? So if we're going to make chloride move such that the membrane potential goes from minus 60 to a more negative number, does chloride come in or does it leave? It must come in, right? See, it's very straightforward. Chloride must come in order to shove the membrane potential towards minus 77, at which point there's no more net movement. Okay. And if you wanted to use that other technique, which is the EMF thing, it's minus 60 minus a minus 77. It's what? 17 plus 17. You put plus 17 here. You're looking at a negatively charged ion. It's going to pull it into the cell because it's going to attract the negative charge. So again, that system works. And whichever way you're more comfortable with, then you should do it. But I want you to understand the basis for why it's doing that. There's some energy that's available to push it in one direction more than the other. Okay? Now, let's see what time it is. I think instead of let me give you a real. So this is a test question. So you thought it was going to look like that, right? Let's see. Let me see if I've got that one that has the questions on it. Oh, it's in the other one. So we still have 7 minutes, okay, so don't go anywhere. Oh, there it is. All right. This is what your test question is going to look like. It's the same problem. It's the exact same problem. You have to be able to ignore the crowd. You can guess when this question was written if you read the question. Okay, the only thing that I need to tell you differently is at body temperature, that number is going to change, right? Because the little conditions are, it is the collection of all those variables is a little different at one temperature or the other. So at body temperature, that number is not 58, it's 61.5. So let's just do this. And you see if I'm doing it the right way. So out of all that jiggery-pokery, what you need to do is just get rid of everything that doesn't matter. So here's the thyroid cell, okay? And here's the blood, okay? Whizzing by here. And so we know that the blood, the thyroid, the iodide levels now are high. So it's what, 10 micromolar. Don't let those units worry you, okay? And it's 1 micromolar inside. Was that the correct? Is that the way it was? Okay. And I think it was minus 50 millivolts on the inside. This is actually an epithelium, by the way. Your thyroid is an epithelium. And so we're going to open this channel and ask which way will it move, if any. Okay. And so it's outside over in, and they're the same units, so we don't even have to put the units down, right? We're taking a ratio of two things that have the same units. So it's 10 to 1. What's the log of 10? 1. Okay, so that's a hard one. 1 times, and it's not 58, it's 61.5, but it's also minus 1 because iodide has a negative 1 charge. Now, that's in the question, so that's not tricky either. And so it's minus 61.5 is the Nernst potential for iodide. Okay? So we do our really difficult thing. We go, well, here's the VM. Voltage is minus 50. Before you open the channel, you open it, and it's going to move towards the Nernst potential, which is minus 61. So we need to do something to iodide that will bring the membrane potential towards the Nernst potential. And if it's a negative ion, what would that something be? It's going to go in. There you go. Not difficult. Just get rid of all the trash. Okay, we'll stop with that. Oh, sorry. That's where the information for quiz 2, or quiz 1, quiz 2, quiz stops. Ion transport, right there. Thank you. Important.